Welcome to Anthro PYQ series. So today we are discussing about 2025 paper 10 marker questions. The first question is Mendelian and non-Mendelian traits, so which come in 25 for 10 marker. How do you attempt this kind of questions? So simply comparing these two traits in 150 words. So that is enough. So now we are going to discuss. So first you have to break down that question into related keywords. First one is Mendelian and non-Mendelian traits. So Mendelian traits, it has only one type of definition, but non-Mendelian traits are many more are there. So introduce that keywords. Mendelian traits are, those are all followed fixed laws in their inheritance from parents to offsprings. So those laws are law of segregation, law of dominance and recessive, law of independent assortment, law of unit pair. So they are governed by single loci on the homologous chromosomal pair. So what about non-Mendelian traits? Non-Mendelian traits does not follow Mendelian rules or laws while they are inheriting from parents to offspring. So that you have to write. So these are all classified again into polygenic traits, okay, pleiotropic traits, cytoplasmic traits, and epigenetic traits. So these things you have, these mechanism of inheritance we call as a non-Mendelian inheritance. So this is very straight and simple to understand. This is very complex to understand. Complex traits of inheritance. So now, why do both are important for an anthropology student or a biological anthropology branch? So both govern the human variations, adaptations, and disease inheritance pattern from parents to offspring, and even in population genetics, microevolution also. The gene and genotypic frequencies changing from one generation to another generation, that generally we call as a microevolution. The same thing, if it is occurred millions of years, that we call as a macroevolution. So to understand all these things, we need to have thorough knowledge regarding Mendelian as well as non-Mendelian traits. So now we are discussing about, once you come to know or you introduced about the definition of Mendelian and non-Mendelian and their significance, you have to list out the characteristics of Mendelian. So Mendelian characteristics are single loci or monogenic. Monogenic traits means if it is a homologous pair, single loci combinedly, locus, this is the loci, this is allele 1, allele 2. So both are same allele or different alleles, but they follow, that means entire trait controlled by a single loci. So entire genetic trait, so which is expressed from a single loci, that we call as a monogenic or single loci traits. So clear dominance per se, here two contrasting alleles are there, like A and O. It only inherited one allele, another one is suppressed. So this one we call as a law of dominance. So this is how they follow clear-cut relationship of dominance and recessive. We can predict, if we know which one is recessive, which one is dominant, we can predict what kind of blood group inherited by the offsprings. So predictability, that already we discussed. Discrete. So discrete means where we can put persons either or category. So here the loci has A allele as well as the O allele. So the person born with either A allele or allele, but not the medium. Okay. So predictability means we can, here we can say that discrete means we can put persons into either A group people, B group people, or AB group people, and O group people. So there is no blood group between A and O. Okay. Clear-cut separation of people on the basis of their trait is possible in Mendelian. So this is monogenic. So the role of environment is zero. Environment cannot modify this inheritance except some Bombay blood group is there. That is different case. We will see in our classes the exceptions for a Mendelian traits. So if you want to give some examples, you should immediately introduce the examples like sickle cell anemia, cystic fibrosis, Huntington chorea disease. So attached versus free lobe. So these are all the traits follow the simple rules of Mendelian. When it comes to next one, so anthropological significance, you have to write immediately. Mendelian traits are very important for the population genetic studies. tracing of mutation frequency in a population from generation to generation, understanding the heterozygotic advantage, that is selective advantage. So helps reconstruction of migration pattern of the parents, like West Asia people migrated into India or Central Asia people migrated into India. So our ancestors we could trace by using the simple Mendelian laws, because they never deviate across the generation. They follow fixed pattern of inheritance. So basis for calculating, so genetic distance between the population, how far they were separated, that also could be estimated by using the Mendelian laws. So next one, non-Mendelian laws. So those are all classified into polygenic. So multiple Lokis. This is the Loki 1, this is the Loki 2, this is the Loki 3. So when single Loki, single trait relationship is Mendelian law, multiple Lokis together, they produce a single trait. For say skin color, if you take, so skin color is the result of 3 Lokis. Okay, so a skin color is a trait. So the trait is controlled by 3 Lokis on the DNA or chromosomes. So that's why poly means many Lokis are involved in the trait inheritance, that we call as a polygenic inheritance. So here, based on the variation in the allele, a person from a pure white to so dark, we can identify the persons belongs to different colors. So from one end to another end, we can see different types of people. 50% white, 50% black people also exist, that we call as a mulattoes. So mulattoes, Devonport experiment, it revealed that so these skin color people are continuous from one end to another end. Okay, so here they couldn't follow the Mendelian rules. So next one is incomplete dominance. Here heterozygote shows intermediary phenotypes. So Taise carriers, this is the best example. So heterozygotes means here you can see incomplete dominance. So A and B people on the Loki, one allele is A, another allele is B. So the result is AB. Neither, so this is best example for co-dominance. A is dominant and B dominant. So this incomplete dominance, HBA and HBS. So HBA and HBS both are expressed in a single individual. So that means this is called incomplete dominance. Here also it does not follow the Mendelian rules. So in co-dominance means generally A is dominant over B, O, and B is dominant over O. But A is not dominant over B and B is not dominant over A. So now if a Loki has both A and B, because no one is dominating the other, both are expressed in the trait. The person inherit the AB blood group. That you should remember. So cytoplasmic inheritance means the DNA present in the mitochondria. So mitochondria generally contributed by the mother. So all the genes which present from the mother mitochondria directly inherited by the both sons and daughters. So here there is no genes coming from the father. It also deviated from the Mendelian rules. This is cytoplasmic inheritance. So epigenetic inheritance means epi means more environmental impacted. So more environmental impacted modification on the gene expression without changing the DNA sequences. So just like DNA methylation and everything, we can say it is the example. So now you can see the characteristics. So you can see the characteristics of Mendelian. Those are all single Loki, and this non-Mendelian impacted by environment, that is epigenetics, and it is controlled by the multiple Loki. So phenotypic variations, you can see discrete. Discrete means either or kind of category. Continuous, we cannot put a person into either white. See, if you take the mulatto person, we cannot put him under the pure white or pure dark. So he is intermediary. So from one end to another end, all the people are existed. So this is quantitative. How much each Loki contribute? How much each Loki contribute for the trait expression? That we will, how much we will measure it. Okay, here qualitative, either dominant or recessive, which is going to be expressed. The quality we are able to identify. So classical examples, already you know, sickle cell anemia, Huntington chorea, cystic fibrosis. So skin color, height, So, blood pressure, AB blood groups, all these things are non-Mendelian examples. So if you take the predictability, so here we can accurately predict because the laws follow rigid rules. Here complex, so it varies with environment, it varies with the gender, it varies with the other factors. So we cannot predict what kind of things coming into the children. So now inheritance pattern follow clear-cut dominant and recessive rule. Here polygenic, cytoplasmic and epigenetic rules are followed. So anthropological use. So Mendelian rules are heavily used in the population genetics and migration pattern. So when it comes to non-Mendelian, which is very useful to understand the human diversity, adaptation, and health, epidemiological anthropology, etc. So this is the clear you have to write. So conclusion, Mendelian traits gave foundation for the genetic studies because it has high predictability, monogenic, and discrete in nature. Non-Mendelian traits which give us knowledge about understanding the human complexity in the form of polygenetic and epigenic and continuous traits. So both are important. So this is how you have to write the conclusion. So for me, I have taken these various sources. So even this is well explained in our material. You already know, I made already 7 volumes of anthropology material. So this year I am going to release the books in the market also. So this is all about the question number 1. We are going to continue this series. Every day I will introduce one POQ in anthropology.